While X-ray powder diffraction is an extraordinary tool for compound identification, it really functions like a fingerprint. And just like fingerprints, it is widely assumed that each one is unique, but each person likely has dozens or hundreds of individuals whose prints are similar enough to generate a false match. Diffractograms are a little more discerning than fingerprints, but do have a similar issue. For instance, a number of inorganic salt compounds have the same packing scheme, and almost all pure metals fall into one of two packing schemes, which leads to extremely similar peak distribution patterns. If the atoms are of a similar size, then the diffractograms may need to be differentiated with the inclusion of standards. I would like to take a moment and differentiate some of the terminology to avoid any confusion. Isostructural molecules are compounds with similar bond distances and angles, which are extremely common among compounds that have similar atom counts and electronic states. Water is isostructural with hydrogen sulfide, methane with the ammonium ion, and all halides that exist in a diatomic state are also isostructural, along with atmospheric nitrogen and oxygen. Most metallated porphyrin molecules are also isostructural. However, what we're discussing in this discussion are isostructural phases in the solid state, which require that the molecular packing structures are largely identical. This usually means the same space group and similar unit cell sizes. Isostructural phases will have extremely similar X-ray diffraction patterns. In the pharmaceutical industry, isostructural phases are most often to occur between different molecules that have a single atom difference. The chemical difference between a hydroxyl group and a thiol, or a furan and a thiophene, are significant, but crystallographically they're very similar. They will have almost identical bond angles, and both can engage in interactions donating electron density from their lone pairs. Alternatively, monohalide salts such as fluorides, chlorides, and bromides will generally have the capacity to form similar packing structures. It might be a challenge to differentiate between the starting material and a product for a chloride to bromide ion exchange, making for a frustrating reaction optimization. Most of the time, these situations are not serious sources of consternation for scientists characterizing a compound, because orthogonal methods will be applied to spot the differences. However, there is a challenging situation in which a compound can have a broad selection of phases indistinguishable from one another through XRPD, which we will cover in the next topic.